There are currently 7 units that have been ported to OLI. And the most important ones, the most central to pathway analysis, are units 2 through 5, gene interaction pathways and epistasis and metabolic pathways, or assembly line pathways, I should say, gene regulation pathways, developmental pathways, and the development of the organism and development of an embryo, and cancer pathways. Each of these units has 2 tutor lessons. A forward reasoning, where you're predicting what's going to happen in a pathway if you know the underlying properties, and then a backward or experimental component where you're shown data and asked to run the pathway backward to determine what could have led to those observations. So let's take a brief look at the declarative instruction for gene interaction to get an idea of what that looks like. So again, as I said, this is much like the chapter, the text in the chapter of a book that you read, which gives a lot of background information, all very important. And when I show you what the tutor lessons look like, it may not make a whole lot of sense, and that is, of course, because you have not read all of the declarative information, all of the declarative instruction that goes into understanding the lesson. So one of the things that the gene interaction and epistasis lesson explains is how different pathways are affected by in crossing 2 parents with different genotypes that affect these pathways. One of the things you will note is that in the instruction there is often in several of these lessons some questions that are asked as you're reading through the material, just to encourage students to read more deeply what they're learning about, to better prepare them for the tutor lessons at the end of the chapter. This shows an example of what happens as students answer these questions about what they're reading about. If they are correct, the answer will turn green. If they're incorrect, the answer they select will turn red, with a description of why what they've selected is incorrect. After they read the declarative instruction, they will come to the cognitive tutor. So let's take a look at what some of these lessons look like. So here is an example of a cognitive tutor lesson. And this is gene interaction pathways, the forward modeling, the prediction. So the first thing the student does is they read the problem statement, which describes a pathway. The first thing the student does is they summarize the problem. This problem has been largely completed. Excuse me, just to show you what this activity looks like. So the first thing the student does is summarize the problem description. Then they determine the phenotypes for each of the true breeding genotypes described by this system. A true breeding genotype is one that is homozygous for both genes. Then after they do that, they examine crosses between 2 true breeding strains and look at to determine what the ratios would be in a case where you have what's called a dihybrid cross. Again, this will make more sense. It would make more sense if you read the declarative instruction. Several of the genotypes result in the same phenotype. We then collapse those, and as the student is solving the problem, if they don't know what to do, they can ask for help by clicking the help button, and then get more help by clicking this, and eventually it will explain to them what they need to enter here, and this is the answer for that. And in one case you can get brown, and there are 3 of the 16 possible combinations are brown, and in this case that is the only genotype that will give you brown chicken. Then after you do that, you're asked to summarize again more concisely, or all the different genotypes that can result in a yellow chicken. And this explains the reasoning leading up to that. If you answer correctly, it will turn green. If you answer incorrectly, it will turn red. Here we've answered that correctly. And this is, and then after specifying the genotype, you describe what will happen if you have one of those genotypes, and given those. In the experimental version of gene interaction, you are given 3 different strains, in this case 3 different types of cats, brown, black, and gray, and you're asked to cross them to look at the offspring. And the first, what you are given when you do that, you are given the data, and you're asked to conclude something about the genotypes of these different strains of cats. And then after you collect all the data, you can determine what the genotypes are for these 2 genes, and then you go back and fill in what the genotypes were for the crosses, which you did not know as you were doing them. So you elaborate all of this as a result of these 3 experiments you performed. And then after you do this, you can guess as to how those 2 genes were interacting in the pathway. So in the second topic, gene regulation, students read a problem statement describing how a particular gene is regulated, how it's turned on or off. They summarize. The first thing they do is they summarize the system, and then they look at, again, the underlying parameters. In this case, they look at several genotypes, several different genotypes, several different mutations. They first characterize those mutations, and then they, with the menu, they select what the impact of that mutation is on the system. And then after that, they determine whether the system would be functioning correctly. In this case, when a particular molecule that turns the system on or off, an external molecule called an effector, comes in, if the system is not functioning properly, then it would always be off. So this is the forward reasoning version of this lesson. The backward experimental version of the lesson, the student is given a system with 3 genes that they can manipulate, and they need, just as in gene interaction experimentation, they perform several crosses, look at the data, and based on the data, they draw inferences about what kind of system, what kind of regulatory system this is, and what the roles of the specific genes are. In the developmental genetics lesson, which introduces signaling pathways, the student again is shown exactly what the underlying properties are and is asked to trace through the individual states of the signals within a signaling pathway. And in some cases, there are mutations that break the pathway so that the molecules in the pathway, the signals, are permanently off or permanently on based on that mutation. In the backward version or the experimental version of this lesson, they're given a set of strains, and by performing crosses, by performing experiments, they learn about how the genes are ordered in a pathway and whether each step in this pathway is activating or inhibiting the next step in the pathway. In the cancer pathways lesson, again, there are a number of mutations that are described in signaling pathways that are involved in the cell cycle to turn the cell, to stimulate cell proliferation, cell growth, or there are also pathways in the cell cycle that turn it off or kill the cell if it's broken, if there are mutations that make it unable to function. So this allows a student to look at a cell with a collection of mutations to determine ultimately whether there is a change, an increase or decrease in cell proliferation that might lead to cancer. After they've done the predictive forward reasoning, then there's an inductive form of that lesson, an experimental form of that lesson, where the student is told that you have overactive, for example, overactive or underactive, in this case, overactive cell proliferation, and they're asked what change, what mutation in this particular signaling protein could have caused that. And ultimately, the student is asked if they were trying to design a cancer treatment for a patient who had a cancer with a mutation in that gene, how they would treat that particular, this particular mutation. Modern cancer treatment involves making a cocktail with a collection of drugs that treat different mutations in a particular patient's cancer. So in this case, this asks the student to think about the different ways that scientists design cancer treatments customized to a specific patient. Well, I hope that that this wasn't all too overwhelming. And again, all of this, we have shown that while this lesson, while this tutor was designed for college biology majors, primarily at Carnegie Mellon, but other places around the country, we have introduced this very successfully at several high schools, and high school students in AP biology and honors biology are very capable of learning this, and the learning gains that we have seen are comparable to the learning gains that we've seen in biology majors at several universities. So we are optimistic that very large numbers of students will be able to learn the logic of pathway algebra and develop a deep understanding of biology, biological reasoning, and what biologists do as a result of that.